Hello everyone. Thank you for attending my virtual presentation of mercury methylation and demethylation in paddy soil with and without. So, methylmercury is a dangerous neurotoxin that contaminates rice grain. As most of you are probably already aware, even extremely low levels of methylmercury can cause irreversible neurological impairment, particularly if the person is being exposed in utero before they're born. And this is not a small pathway of methylmercury exposure to the global population. In fact, today, rice represents almost 10% of the global intake of methylmercury. And in some areas, it can represent the majority of the methylmercury that people eat. It can represent more even than the fish that people eat. So this is not a minor, this is a significant public health concern. And anything that we can do to reduce the amount of methylmercury in rice grain is going to be an important public health intervention addressing this emerging concern. So where's that methylmercury in the rice grain actually coming from? Well, it's coming from the soil. Methylmercury in rice grain originates from methylmercury that is created in the soil around the plant's roots. Methylmercury is created from inorganic mercury, from a less toxic and less bioavailable pool of mercury species collectively known as inorganic mercury. And the process of methylation is carried out by a taxonomically broad association of soil microbiota called mercury methylators. Now, methylmercury can also be degraded or demethylated back to the inorganic form by a different group of microorganisms known as mercury demethylators. Now, since these are both biotic processes, it's, as you might expect, that the activity of the plant itself alters the soil biogeochemistry in ways that have the potential to affect methylation and demethylation in rice paddy soil. But unfortunately, the majority of the work on methylmercury biogeochemistry has been carried out in natural systems, and it's not really clear whether methylmercury production or degradation is more important in controlling soil methylmercury levels in rice paddies. Better understanding of this very fundamental question could inform efforts to breed plants that alter the biogeochemistry of the soil around them in ways that might control the methylmercury burden and thus control the methylmercury in rice grain. So we took a simple first cut approach to investigating this question. We maintained California rice paddy soil in shallow rhizoboxes with and without rice plants. These rhizoboxes allowed us to access the submerged rhizosphere. For simplicity, I've left out the flexible panels that we used, flexible vinyl panels that we used to access the rhizosphere without also introducing oxygen, but we did have an oxygen introduction control strategy in this system. So we waited until the plants had achieved tillering, the peak of vegetative growth, after which we injected stable isotopes. Two different types of stable isotopes, mercury 200 and methylmercury 201. These isotopes allowed us to track the formation of methylmercury from the mercury 200 tracer or the degradation of methylmercury using the methylmercury 201 tracer. We incubated these patches of soil for 8 hours, after which they were collected, and we used the transformation rates, the amount of each tracer that had been converted to the other mercury species, in order to calculate the methylation and demethylation potential rates. Just a quick note, why are these called potential rates? Well, they're called potential rates because the bioavailability of the tracer for methylation is different from the bioavailability of the methyl mercury and inorganic mercury that's present there naturally. So these are not absolute rates. They couldn't be used to calculate the absolute methyl mercury production capacity of this system over time, but they do give you a window, a snapshot into what's going on an ecological level in this system. We also analyzed the same patches of soil, the same plugs of soil, for the methyl mercury concentrations and inorganic mercury concentrations. And those are the amounts of methylmercury and inorganic mercury that were present in the system aside from the tracers. These are the background levels of methylmercury and inorganic mercury. So what did we find? Well, looks like the methylmercury concentrations are significantly less variable in the soil with plants. Although there was no treatment level effect, the the variability was significantly reduced in the planted sediments, and it's notable that that reduction occurred mostly in the uppermost values. So if the uppermost methylmercury concentration had been sort of chopped off in the planted sediments. We also observed that there was a similar pattern for the methylation potential rate, abbreviated as Kmeth. Again, no treatment level difference, but significantly lower variability. And again, that reduction in variability is almost all in the form of a reduction in the uppermost levels. So it's like the plants are somehow reducing the top levels of methylmercury production and the top levels of methylmercury concentrations in the soil. Now, you might be wondering, well, is that just because there was less of the inorganic mercury substrate? And no, that wasn't the explanation. In fact, there was no treatment level difference. There was no difference in variability. The microbes in the planted and the unplanted boxes were both working with the same amounts of inorganic mercury to start with. So something is being altered in terms of methylmercury production. And something is being altered in terms of the ability of the soil to sequester methylmercury. So we moved on to examine this data on a correlative level. What we found was a little bit surprising. There was only a significant relationship between mercury methylation potential rate and soil methylmercury concentrations in the boxes that didn't have any plants in them. There was no correlation in the boxes where there were plants. So even though it looks like the plants are doing something to the methylmercury production rate in the soil around their roots, there's no correlation between that something and the methylmercury concentrations. It looks like methylmercury production and methylmercury concentrations have been decoupled from each other in the planted sediments. And this contrasts with what we see in the unplanted sediments, where there's a clear and strong positive correlation between Kmeth and soil methylmercury concentrations. And this is very typical of what you see in most natural systems. So turning to demethylation, what did the plants do to demethylation rate? Doesn't really look like they did much. There's no treatment level effect. There's no difference in variability. There's a lot of variability. There's a lot of spread in demethylation potential rate in both planted and unplanted boxes. But despite this, demethylation does appear to control the methylmercury concentration in soil with plants. There's a clear and strong negative relationship in planted sediments only. So the more demethylation you have, the less methylmercury you have when the rice plants are present. Now, when there aren't any rice plants, there's no demethylation detectable. No correlation between demethylation and soil methylmercury concentrations is detectable. Even though we know that demethylation is occurring in this soil, it doesn't seem to be a controlling factor on the final methylmercury concentration. And this is quite typical of what we usually see in natural systems. So the fact that we're able to detect this relationship between demethylation and methylmercury concentrations in the planted sediments is actually rather unusual. So we believe that the explanation, or at least we speculate that one explanation for this apparent quandary is that the plants are indeed altering what's going on in the soil around their roots, and the fact that those plants were packed in so tightly in those boxes is making this effect apparent. Methylation is an anoxic process, while demethylation occurs in both oxic and anoxic settings. We know that rice roots leak oxygen from the soil around them, and this leakage of oxygen was probably limiting the uppermost levels of mercury methylation rate, was reducing the ability of the mercury methylators to grow and metabolize and to methylate mercury. So this was detectable as the reduced variability in Kmeth that we saw. At the same time, this decoupling of methylation rate and methylmercury concentrations actually allowed demethylation to shine forth, to be detectable as an actual control on methylmercury concentrations in this soil with rice plants, even though the rice plants didn't necessarily seem to be altering the demethylation rate directly. So in conclusion, mercury biogeochemistry does seem to differ between soils with and without plants. Methylation and methylmercury concentrations were both significantly less variable in planted soil. Mercury demethylation correlated with methylmercury concentration in the planted soil, but these demethylation potential rates were not different between planted and unplanted soil. And we speculate that radial oxygen release from the plants explains this apparent quandary. We'd also like to point out, to emphasize that that the planted rice boxes are displaying a different mercury biogeochemistry than most natural systems, and that the unplanted boxes display a mercury biogeochemical pattern that is more typical of your average natural system. We speculate that the soil near plant roots may have a unique mercury biogeochemical cycle that is influenced by the radial oxygen loss and perhaps other processes controlled by the plants. And we suggest that it may be possible to breed plants, perhaps through breeding plants with increased radial oxygen loss, to further decrease mercury methylation or to further enhance mercury demethylation, and thus help control methylmercury grain concentrations in rice and to help address this emerging public health problem. Thank you for your attention. Please do submit your questions early to the question and answer to the Q&A committee so that they can be organized, and I look forward to speaking with you all during the Q&A session.